This is the Wadden Sea, and in this sea, a complex ecosystem of many organisms exists. Together, they make up a beautiful landscape and provide many services. However, this system is subjected to more and more pressures, one of them being the introduction of invasive species. Around the 1990s, Hemigrepsis takanoi and Hemigrepsis sanguineus have vested their claws upon the Dutch shores. Both species, being opportunistic omnivores, predate on a wide variety of food sources, making them excellent invaders. Consequently, their numbers have been increasing. Great, for them. But what about the rest of the ecosystem? To answer this question, we need to gain a better understanding of their feeding habits. So that's what we set out to discover. Our research question was, what is the dietary preference of Hemigrepsis takanoi and sanguineus? Do they compete for the same food sources? As both species originate from the same area, we hypothesized that both would show a similar preference towards animal-based food sources.
Let's move on to the methods. First, we had to catch our crabs, which was 10 individuals per species. For this, we went to a dike just north of Oude Schild, which started out great, but we were not able to find both species, therefore we also had to find our other species at the Mokbai. We selected for crabs that had no specific conditions like missing claws or parasites and ones that were similar in body size. Prey was also selected. It was mussels, snails and algae. The crabs were kept in individual buckets containing a little bit of salt water and an aeration tube. Buckets were covered using aluminium foil to support some sort of safety. For our first experimental setup, we used PVC pipes. Pipes were placed in rectangular buckets, and in each of the 3 pipes a different prey type was placed. The crabs were placed at the center and could freely choose to which prey type they wanted to move. Unfortunately, the crabs were not really doing what we expected. Instead of moving to the prey and feeding on it, they preferred going into tiny spots and hide, probably because they weren't feeling very safe. So we had to think of another experimental setup. This time, crabs maintained in their individual buckets. Inside these buckets, prey types were placed. The crabs were left inside these buckets overnight for approximately 12 hours. Initially, we wanted to weigh our prey types, but due to varying water contents, this was super difficult to do, so we had to think of something else again. And thus we decided to measure square millimeter surfaces, but more on that later.
Based on the result of the t-test and the analysis of the box plot, the conclusion is evident. In the control group, there was not significant change due to water absorption and aeration. The first experiment illustrates a decrease in mussels and snails significantly for both species. The area change of the algae is not obvious, and nearly half of the mussel have been consumed. In the second experiment, after replacing the snail with oyster, only the consumption of the mussel is significant.
To analyze our surface area data, we encountered some challenges because of the light conditions, the three-dimensional structures, and the fact the prey sometimes had holes or dangling parts in some photos. Because of this, the software couldn't always recognize the outline of the prey by itself. We therefore sometimes had to trace the outline of the prey by hand in order to calculate the surface area, which may have led to a form of bias, as it may be less accurate than the software program. To prevent this bias, in the next experiment, the images can be relabeled, so the person tracing the prey is unaware of the exact prey identification, and thus doesn't know whether it's a control, sanguineus, or takanoi image. It is also recommended that in this experiment, prey of similar sizes and volumes are used. However, we didn't have enough mussels to have the same sizes of prey for all experiments.
Concluding from the results, the 2 invasive crab species, they show similar food preferences, namely the preference in mussel, meaning that they presumably have competition over this food resource, should they occur in the same area. And this is something we actually noticed in the field as well, is that the 2 species would barely be present under the same rock. So competition over area, it could be very interesting to look into for further research, to paint a better picture into their future effects on Dutch coastal habitat. And the results of our experiment can give us a better understanding of how this competition worked between the 2 studied crab species works. And this can give insight into what species may dominate the Dutch coast in the future, and also help us better design strategies to protect our beautiful coastline.